Okay, welcome back. Today we're going to go over Chapter 2. And just before I start that, I do want to remind you that we've gone over a little bit of the history of microbiology in Chapter 1 and learned about the basic characteristics of the members of the microbial world and the role of microbiologists in um, understanding human disease. And really this second chapter is all about public health. And so you may be wondering, what does microbiology have to do with public health? But really, microbiologists are critical in our understanding of infectious diseases. And public health departments and officials are responsible for understanding, among other diseases, infectious diseases. And so as such, they really rely on the information that microbiologists provide them about the microorganisms and the microbes that cause human disease. So in this chapter, I need to warn you ahead of time that it's very terminology intensive. It may be that these terms are unfamiliar to you, and so you will need to spend extra time just learning the terms in order to understand the concepts here. But basically, I want to move away from what is a microbe and what are microorganisms and how are they described and characterized, to an understanding of what is infectious disease, because this course is really focused on microorganisms, microbes that are associated with human health and disease. And so in order to kind of set the stage for what we're going to learn about the microbes this semester, you really need to understand infectious disease. Okay, so this chapter, in this chapter, I'll be going over those concepts of how microbes fit into this picture of the whole picture of an infectious disease and what we understand about it. And there are some key things listed in the objectives that I want you to be sure to be able to do as far as the learning outcomes for this chapter. So pay attention when you read the chapter to taking notes on these terms, these concepts, and these topics. Okay, so once we define infectious disease, I'm going to move into public health and epidemiology, which is the kind of the second half of the chapter. And so I'll be asking you to learn and understand some key terms in this section, as well as how microbes fit into our understanding of infectious disease. Here's a list of terms that are really key and fundamental to your understanding of this chapter, but there are more that may be unfamiliar to you, so as soon as you come upon a term that's not familiar, be sure that you're taking the time to look it up and understand what it means. Okay, so today I'm really focused on infectious disease, but infectious disease is a subset of disease, and disease by definition is the damage that is caused to an organ system or a body part that can be recognized by a set of signs and symptoms. And in infectious disease, often diagnosis begins with an evaluation of these signs and asking the patients about their symptoms. However, not all disease is caused by a microbe, and so that that's really what distinguishes infectious diseases, and that's what we're going to focus on in this class, from disease in general. Okay, so infectious disease caused by a microbe, whether it's viral or a microorganism, bacterial or fungal, or parasites such as a helminth or a protozoa, all are all cause damage and disruption of the normal function of the human body. And they can be transferred from one host to another. They're not all contagious or communicable directly from human to human, but they can be transferred from one organism to another, or from a reservoir, an environment, to us as humans. So I always want to balance in this course your understanding of microbes in their pathogenic form, in which they do damage and cause disease, with those that are really beneficial to us. And so normal microbiota, we're now understanding from the Human Microbiome Project that it really is an indicator of the health of our body. And so we need to understand that many of these members within the same group that are pathogenic, there are counterparts that are not pathogenic, that are actually commensal or beneficial to us. And that's what we call normal microbiota, and it includes viruses and bacteria and archaea and other eukaryotic microorganisms. So there are different relationships that microbes have with our body. Some of them are mutual, in which both our human body, is benefiting, as well as the microbe. So clearly the obvious benefits that microbes have of inhabiting and colonizing our body is that they are able to find an environment in which they can get food and have space to grow. So colonization is this description of microbes and their growth in our body. And microorganisms need to be attached to a body surface before they can colonize or replicate and reproduce. Viruses have to get into our cells before they can replicate. So some key characteristics that are necessary for a microorganism to attach to a surface on our body are that they have to have factors, proteins on their surface that allow them to attach, and those are called adhesins. They have to have the food available so that once they attach, they could grow. So our body is a rich source of food no matter where, but especially the gut, because we are constantly feeding our body and the food is going through our gut. So we have lots of bacteria that are normally found in our gut because they find that the environment is really conducive to growth there. And they have to find space in that environment in which to attach. If there are already normal microbiota in our gut that are attached to the surfaces of the lining of the epithelia lining the lumen of the gut, then a pathogen, when it comes along, will not have the right environmental factors for it to complete its colonization. So it has to compete and find that space among the normal microbiota in order to successfully cause disease. And then there's susceptibility to pathogens. So our health, the state of our health, whether we're immunocompromised or healthy, will influence whether an opportunistic pathogen can colonize. A primary pathogen often has other virulence factors that allow it to colonize despite us being healthy. So here's an electron micrograph of E. coli, which is the labeled there. It's the rod-shaped spherical gray dash-shaped cells in the electron micrograph, and they are actually attached to the surface of human tissue in the gut. And you can see the microvilli around them. And this is really key. We have E. coli is a really interesting case because this one species has many subspecies, many strains, which are all the same species but differ genetically in slight ways. And so E. coli that is actually a normal microbiota. It's found in our gut in very small amounts, but in our gut of virtually everyone. And yet E. coli can be a pathogen. So one thing that we're going to really I want you to focus on this semester is to understanding the differences between a non-pathogenic microorganism and a pathogen. And they often have very similar genetic makeup. So they're the same species, like the E. coli that is in our gut normally. That's mutualistic because it actually uses the nutrients that we take in as we eat and produces vitamin K for us. So an important clotting factor that we couldn't live without. And so we benefit from E. coli being in our gut, but when we ingest food contaminated with E. coli, a different strain like O157, we get food poisoning and we get diarrheal symptoms. And so there's a genetic difference in that the pathogenic form has additional factors we call virulence factors that allow it to cause damage to our healthy gut. So a pathogen is any bacterium, virus, any microorganism or microbe that can cause damage, cause disease. And pathogenicity is the ability, so that genetic makeup, those virulence factors that give the ability of a microorganism to cause disease or a microbe to cause disease. A parasite is the way that we describe all pathogenic microorganisms or microbes because it's able to harm the host, really. Normal microbiota can colonize the host, but they don't harm it. And a parasite or a pathogen, which all pathogens are parasites, are ones that are harming the host. And here we have an example on the left of a type of yeast infection on the skin. So that's an ectoparasite or a parasite on the outside of our body, whereas on the right we have an example of a helminth that can cause infection inside our body, and that's called an endo or inside parasite. So most pathogens that cause real damage and illness are endoparasites. I mentioned already that pathogens differ from non-pathogenic microorganisms in that they have certain factors that enable them to cause disease. And so we can actually measure the degree or the severity of the disease, and that's what we call virulence. So more highly virulent microorganisms, things like Ebola or Marburg viruses, have a very high death rate or mortality rate. They make us very sick, and a lot of people die from that infection. Virulence factors are the traits or the genes that encode the traits that make this microorganism a pathogen and able to cause damage and disease. The more factors that a single pathogen has, often the more virulent it is. And we'll learn later at the end of the semester, we'll learn about pathogenesis and some of these virulence factors and how they influence the ability of the microbe to really evade and get across many of the barriers of our immune system in order to cause damage. So the way that we measure virulence is by either measuring the death rate or measuring how many people get sick. The lethal dose is the death rate, and it can be, if there's an experimental model for the infectious disease, a mouse model or other animal model, then we can actually measure it by exposing a number of animals to the pathogen and then seeing how many of those pathogens, for example, the bacterial count, how many colony-forming units in bacteria or how many plaque-forming units of a virus are required to kill half of that population of experimental animals. In a similar way, public health officials can study a population that's been exposed to a pathogen, or in an experimental system study animal models for that disease and figure out how much of that pathogen is needed to cause symptoms in half of the animals that are tested or half of the people that are studied. And this graph is showing in the blue the lethal dose of one pathogen called agent 1 versus the lethal dose of another. And as the curve shifts to the right, you can see that the dose in order to cause death is much higher. Okay, so all pathogens must be able to infect. They have to get into the host, but they don't all cause disease in every single host they infect. And this is partly because of our immune response and partly because of the nature of the pathogen. Infection, by definition, is when a pathogen enters or begins to grow in or on our body, as if we're talking about human hosts, which is really the focus of what I'm talking about this semester. It may go unnoticed. An infected person can be a carrier and spread to other people. In the case of cholera, which is spread by the fecal-oral route, so the feces can expose other people to it, even if that person is not showing signs and symptoms of disease. Primary pathogens are those that have the virulence factors that enable them to breach the defenses, the immune system, and the barriers that our immune system has in place of a healthy person. Some examples are Shigella flexneri, which causes a diarrheal infection called shigellosis. Opportunistic pathogens, on the other hand, tend to be less virulent, and so they can't cause disease in a healthy person, but if given an opportunity, they can cause disease in a compromised host, so where their immune system is weak or where a situation arises, somebody undergoing surgery, or if there's a perforation of the gut, those normal microbiota can get into areas of our body that are normally sterile, free of microorganisms or microbes. And once they get into that area, they are capable of colonizing, and by colonizing, causing damage to the host. Pathogenesis is what we'll study later in more detail in the semester, but it's really the pathway that that a pathogen takes in its, and the steps it takes to cause disease. And it can be, microbiologists have kind of defined these steps in the order that they must occur, and that we've found that all pathogens take. So there has to be first attachment before disease can be caused, and then immune avoidance. So that means either a barrier has to be breached, or the microbe has to hide out from our white blood cells that are responsible for clearing foreign foreign organisms such as microorganisms from our body long enough. It may not be forever that the immune avoidance takes place, and it's usually not. But it's a temporary time when that gives the microbe enough time to colonize and reproduce and cause damage before the immune system is able to clear it and protect our body once again. So on the end of the pathogen, these are the immune avoidance can be achieved through changing its appearance, donning different disguises, secretion of signals that mimic host signals, so trick the immune system into thinking that this is not something foreign, that it's okay for it to be there, and for signaling apoptosis or killing of the immune cells. And then, of course, just like normal microbiota, any pathogen has to find a way to obtain the nutrients that it needs from the host. And iron is really a challenging nutrient to get, but that most pathogens require. And that's because there's really no free iron floating around in our body. Instead, it's bound up by lactoferrin and transferrin, and the microbes have to have a way of sequestering that iron, either by getting it from, example, red blood cells, by lysing the red blood cells so that they release their iron, or by having molecules, secreting molecules and producing molecules that do a better job of binding iron than our own human lactoferrin and transferrin do. So once the microorganism has attached and it's getting the nutrients it needs, it can colonize and reproduce. Sometimes some pathogens then go on to invade, but some just colonize and start producing toxins that cause damage, or just simply the colonization causes damage. Invasion to an area of the body that is not colonized by normal microbiota is a key step in many pathogens. The ability to invade is really, again, due to the genetic makeup of the pathogen, these virulence factors. And so, for example, Streptococcus pyogenes, the form that is flesh-eating bacteria, produces a toxin that rapidly degrades the components within the extracellular matrix of our tissues, and kind of breaks down the tissue and destroys the integrity of our skin. And that's by toxin production. So really the most severe diseases are those caused by pathogens that produce toxins. And these are exotoxins. They're secreted out away from the bacteria that have colonized our body and have an effect on the cells or the tissue, locally or systemically. So here are a couple of electron micrographs just showing examples of invasion. On the left, there's bacteria in the middle vacuole. There are bacteria that are being taken up by gut epithelial cells. So that's one way that microbes can hide away and invade and get across the barrier of the epithelia in our gut, is by inducing the cells along our gut to take up the bacteria. And then Shigella actually does that, as well as it is able to get into from the basal surface to the gut epithelia and remain within the cytoplasm of those cells. So in the fluorescence microscopy image on the right, where you see the red is actually the cytoskeletal elements, the actin that's in the host cell, and the green is the bacteria, in this case Listeria, which, like Shigella, can hide within the cytoplasm of host cells. And so once they're inside a host cell, they're basically hidden from our immune system, because all our immune system sees is that host cell, not the microbe. So there are a variety of definitions for infectious disease. If we get a primary infection, that's an infection of a previously healthy person. A secondary infection is when we're already sick with something, we get sick with another infection. And so these are usually opportunistic pathogens that cause these secondary infections, because they're not, their virulence is not great enough to cause a primary infection. You can also get an infection from a variety of sources. You can get it from your environment, from what you eat or drink, from a person that's already sick, but you can get it from also a healthcare worker, and there's a special term for that, and it's a key study within public health is to understand these iatrogenic infections, because we want to limit that as much as possible. And hand washing is really key, because we carry so many of these microorganisms and the microbes on our hands, because we're using our hands for so much of the work that we do with our patient. And when a patient comes into the hospital with one infection, or comes into the hospital for, say, surgery, and is perfectly healthy coming in, but then during the hospital stay gets sick, we call that a nosocomial infection, or a hospital-acquired infection, HAI. And MRSA is really one of those methicillin-resistant form, a strain of Staphylococcus aureus bacteria that's resistant to a variety of antibiotics as well as some disinfectants, and we call it MRSA, or methicillin-resistant Staphylococcus aureus. This is one of the key examples of great concern in hospitals today, as well as other antibiotic-resistant infections that are acquired during the hospital stay. And really, it's the environment of the hospital that promotes this. You have sick people, you have people that are taking antibiotics. The presence of the antibiotics influences the environment that the bacteria are growing in, and so only the ones that can survive in the presence of the antibiotic are the ones that persist in that environment. Outside of the hospital, you don't have as much of that, because you have a mix of, for example, soil bacteria in our environment that greatly dilute the presence of these drug-resistant microorganisms. Some infections are very simple and quick, and our immune system is able to efficiently eliminate them, whereas others are more complex and long-term. Something like strep throat or the flu, you get the infection, your immune system clears it. It's called an acute infection. You have symptoms, the symptoms go away, the microorganism or microbe is no longer in your body. End of story. But in the case of more complex infections that are chronic, either the symptoms are acute and then go away and then appear later, or they can develop gradually over weeks or months or a lifetime. Tuberculosis, also called the wasting disease, it is caused by a bacteria called Mycobacterium tuberculosis, and if gone untreated will result in death. And this is a chronic, slowly, gradually over the, if untreated, about a couple of years will take to slowly cause such severe symptoms that the body will not be able to survive. Subacute infections are also, the symptoms take longer to appear, but they are a little bit more quick than chronic infections. And latent infections, things like HIV, where it's actually what we call a slow chronic infection. It takes years for it to progress to AIDS or acquired immune deficiency syndrome. Herpes, for example, you can have outbreaks, so the virus is always in your body, it's never cleared, so it's a chronic infection, but you don't always have symptoms. There are periods of convalescence and periods of infection or acute episodes. So we can define the stages of disease and divide them into 5 basic stages. There's, and look at the graph here in this figure. So here on the y-axis are the number of microbes. So how many bacteria are present or how many virus. And then on the x-axis is time. So when you get exposed to a pathogen, there's first, you have no idea you've been exposed. There's an incubation period, no signs or symptoms. And signs being something that can be observed by a nurse or a doctor that's examining a patient, whereas the symptom is only felt by the patient and has to be described. So you have to ask certain questions in order to understand what the symptoms are. The second phase is a prodromal period where there are mild kind of general signs or symptoms. Oh, I'm tired, oh, I'm achy, but not defined, not unique to that infection. And then you get sick, your period of illness. That's when, as you can see by the red line peaking here, you're getting the most severe signs and symptoms. Your body, or with the help of medication, may then enter a period of decline where you're getting better, feeling better, less signs and symptoms, and finally, in the case of an acute infection, you'll have a period of convalescence where the microbes, as you can see, are cleared completely from your body. In the case of a chronic infection, the microbes won't go down to 0. They'll stay at a very low, sometimes undetectable level in the period of convalescence, but then rise again during an acute episode of a latent infection, for example. And I've already described these, but I just have them written out for you to make it clear. And then another way of looking at these phases is to look at the immune response as well as the number of microbes. So why do we have a peak of the number of microbes, as you can see, during the illness phase, and then why does it decline or decrease in the decline phase? It's because our immune system takes time to kind of ramp up and get rid of that microbe and respond to its presence in our body. And so while the microbes peak in the illness phase, once our immune system kicks in and peaks in the decline phase, it's really getting rid of those microbes, and it stays elevated through the convalescence phase until in the case of an acute infection, the microbe is completely cleared from our body, or in the case of a latent infection, the microbe is kept at low enough numbers, and our immune response will then wane. So, as I mentioned, all of this is leading up to epidemiology, the study of disease, but also infectious disease. And I'm just going to talk about the traits that are important for infectious disease. But epidemiologists rely on the information that microbiologists provide about an agent, a pathogen, in order to make recommendations for public health policy and for treatment of infections and infectious diseases. So we already talked about the agent and the pathogen being a pathogen that has virulence that can be measured and is inherently genetic trait in the genetic traits of that microorganism or microbe. The other factors that epidemiologists need to study in order to understand and make policy recommendations is the host. Who is the host? How many hosts are there? Where are the hosts found? And the reservoir. That pathogen, where is it found? Is it only in humans and it just transfers from transmits from human to human? Or is there another animal that can get infected with this pathogen? Or is there an environmental reservoir, water or food, that is a source of this pathogen? And the way that epidemiologists or microbiologists help epidemiologists to understand these, at least the agent, the causative factor of that disease, is by using Koch's postulates. So Koch was a scientist and physician that was interested in actually anthrax, which is a bacterial infection in animals. And can also cause death in humans and infect humans. And he developed a series of 4 basic steps in order to identify a microorganism as the cause of an infectious disease. These 4 steps are that the microbe must be present in every case where the individual is sick. The organism must be grown in pure culture, so it has to be clear that you're studying only one microorganism and that that microorganism is present in each case of the disease. And then the same disease has to be produced in a susceptible experimental host. So using that pure culture of the microorganism, introducing it into an experimental animal, and then again looking for the signs and symptoms, if it's death, looking for the death of the animal, and then being able to harvest and grow from the experimental animal the microorganisms that were found in the initial case. So the microorganisms are grown as we will grow microorganisms in the lab, either in liquid or on a solid media surface. And then once there's in pure culture, they are studied under the microscope. And that's really this appearance under the microscope, the microscopic morphology, which we'll talk about later, and learn about in lab, is really a key factor in identifying microorganisms or pathogens. But it's not the only piece of information that can be used. So many microbes have similar appearance under the microscope, and so further testing must be done in order to definitively identify the pathogen. Once the cause of disease is understood, epidemiologists are also interested in knowing, well, how prevalent is this disease? How severe is this disease? And so they study morbidity, which is how many people get sick out of those being exposed, and mortality, how many people that got sick die from a disease. And actually the CDC, our national public health organization, publishes a weekly report where you can find out about new and emerging infectious diseases, called Morbidity and Mortality Weekly Report. So epidemiologists deal a lot with statistics around these measures of infectious disease. And also in making recommendations for the prevention of disease, they have to understand not just what is the host, so go back to this triangle relationship. There's a disease here in the red that we see is characterized by an agent or a pathogen. The host that we're most interested in is the human host, but there may be other hosts, and the environment or reservoir. Okay, so this relationship, understanding all three components and identifying them is important, but we also need to understand, in order to make recommendations, how the agent is transmitted from its reservoir in the environment to a host, a human host. And that's really what we define as disease transmission. So an infection cycle, while part of it includes the damage caused during disease in a host, part of it also includes how it's transmitted to a new susceptible host, because a microbe can't survive if it can't get to that next new susceptible host. And that's really how we've successfully eradicated infectious diseases like smallpox, which is caused by a virus that we were able to break that cycle of transmission with vaccination, and so the virus eventually couldn't find another susceptible host, and we no longer have to worry about smallpox as a possible disease because it's been eradicated from the human population. Modes of transmission can be very direct, human to human, very simple, or complex, involve lots of other environments and organisms as hosts. So direct contact, shaking a hand, sexual relations, sneezing, coughing, those kinds of things are direct contact transmission. Indirect transmission is the pathogen gets to a surface. That surface then you are exposed to, maybe a door handle or a medical instrument, a pair of gloves that have been used for more than one patient. It can also be through food and water, so foodborne infections, or the pathogen can be carried by a completely different organism. We've heard a lot about Zika, and Zika is carried by mosquitoes. And that's what we call vector transmission, vector-mediated transmission, when it's another organism that carries the pathogen to you, and the mosquito, when it's doing its feeding, actually crosses that protective barrier of our skin and regurgitates that pathogen right into our bloodstream, right where we're susceptible. So disease transmission, however the pathogen enters our body, is usually how it exits. So if we get that pathogen by eating or drinking, so the oral route, then it exits and is transmitted to another person by the fecal route. Okay, skin through our epithelial surfaces is transferred skin to skin. Um, Respiratory will be acquired by maybe breathing in some droplets from someone sneezing or coughing, and then as part of the symptoms, the signs, sneezing and coughing that we have when we're sick will then be the way that the microbe exits and finds another susceptible host. Urogenital, so through mucosal secretions that are in the genital and urinary tracts, and this can be through sexual transmission or exposure to those surfaces if they're not kept clean. And then parenteral, which is through injection directly into the bloodstream. That's like the example of Zika and how the insect bite transmits the Zika virus to our bloodstream. But it can also be through dirty needle use of IV drug users. So these infection cycles can be quite simple or quite, in the case of here in the middle, the direct transmission, or quite complex involving other hosts and other vectors or means of in which the pathogen is transferred to us as a host. Sometimes we are the accidental host. In the case of West Nile virus, really the primary host is birds, but we accidentally get exposed through insect bites, mosquito bites. Or sometimes we are the primary host. In the case of influenza, it's person to person. It doesn't involve any of these intermediary steps. So reservoirs of infection, we've already touched upon. They can be an animal or an environment. In the case of the bacteria that's transmitted, that causes Rocky Mountain spotted fever or Lyme disease, ticks are the ones that are both the reservoir and the means of transmission, the vector. And a person who is infected but doesn't have the signs and symptoms of a disease can be a carrier, and I mentioned that before. And that can be, for example, in cholera, which is a diarrheal disease where the bacteria is ingested through contaminated water. Some people don't get sick, and yet the bacteria is still in their feces and can then, if there are not public health systems to have clean water in place in that environment, then can spread to another person who is exposed to the contaminated water. And finally, when there are animals involved in the transmission cycle, we call those zoonotic diseases. And that's, for example, West Nile virus, also the plague, Lyme disease. Usually the Borrelia is in deer or other animals, not normals. We're kind of the accidental host as humans. But if we're in that environment where those ticks are, that they've been feeding, and they've been feeding on deer, and they've acquired and now carrying the Borrelia burgdorferi, then we can get sick with Lyme disease. Finally, I want you to understand some key terms that epidemiologists use to describe the severity or the spread of disease and how many people are affected by the disease. So endemic is a local community that's affected by a disease from year to year. So it's always present in the environment in that geographic site, and people are always getting sick year to year from that disease. Epidemic is when there's an unusual increase in the number of cases that are present in a community in a short period of time. And pandemic is when an epidemic spreads worldwide. So we had an epidemic of Ebola in West Africa. It did not become a pandemic, although public health officials were keeping an eye on that because they were worried about pandemic spread. However, we've had influenza, H1N1 in 2009, be a pandemic and grow to become a pandemic infection. And the flu we know is endemic in our region. We always get the flu from year to year, but the pandemic is different in that that same strain that started possibly in Mexico spread from Mexico to everywhere in the world, almost everywhere in the world. So and this is really a key concern in our current age where the world is quite small because it only takes a matter of hours to travel from one end to the other. There are some factors that the host has that will make it more or less susceptible to disease. So age is one of the biggest factors. The very young and the very old are more susceptible because their immune system is not as strong or either not developed yet fully or weakening in old age. Also, there can be a genetic background that of the host that make it more susceptible to certain strains of bacteria. They're better able to bind because we have that receptor, whereas other people don't. So in the case of HIV, there are some people who are immune to HIV because they don't have the CCR5 receptor that the HIV needs to get into our host cells. And so in very rare cases where these people don't have that receptor, even after being exposed to HIV, they don't get infected and don't get disease. And then just kind of normal hygiene behavior, whether you're malnourished or not, your kind of occupation, are you constantly working with sick people, as you will be, and your underlying health, are you taking some immune compromising medication, or are you pregnant, in which your body naturally changes to immune compromised state. And then I'll let you look over. These are just an overview of the biosafety levels we'll be working with in group 1 in the lab, so non-pathogenic, but there are these definitions in place for the safety of people who are working in the field of epidemiology and microbiology and trying to study these pathogens. And so there are much more serious precautions that you have to take for a group 4 pathogen like Ebola than a group 1 pathogen like E. coli. I'll let you explore the emerging or re-emerging diseases on your own. If you have any questions about any, please bring them to the attention, and we can discuss them in class if we have time in lab. And then finally, I just want to bring your attention to some questions that I picked out among those that are in Chapter 2 that you should focus on for preparing for the test. And of course, use the study guide as well.